Hello class, welcome to Microcosm of the Chattahoochee. My name is Jonathan Clark from EVSP 416 General Ecology. This is my Microcosm project on May 25th, 2025 with Dr. Kristen Przyborski. Again, this is Microcosm of the Chattahoochee. Microcosm overview. This microcosm was done to study the importance of autotrophic species and heterotrophic species in aquatic environments. This was also one to analyze the relationships between the different aquatic organisms. I hope to learn how energy is moved through an ecosystem and why each biological niche is important. To understand this presentation, it is important to keep track of the differences between my two microcosms. Yes, I did two of them. Additionally, it is important to understand the different factors surrounding my water sources. I will go over this in slides 4 and 5. Hypothesis. I have two hypotheses. Number 1, water quality and clarity will improve in 8 weeks as sediments settle and plants and animals sequester nutrients in the water. And number 2, plant growth will increase with access to sunlight and an abundance of nutrients present in the water. Methods and materials, site location. The water source for my project was the Chattahoochee River. It was collected on River Road in Omaha, Georgia. Omaha, Georgia is in the southeastern United States and is part of the temperate broadleaf mixed forest biome. It is part of the southeastern floodplains ecoregion. The climate of this region is humid subtropical. Some of the major impacts of the Chattahoochee River include urban development, energy infrastructure, and agriculture. Urban development. There are about 169 industrial and municipal facilities that discharge wastewater into the Chattahoochee River. Stormwater pollution is the primary cause of poor water quality, with an estimated 500 industrial sites surrounding the Chattahoochee River Basin not complying with clean water laws. Energy. There are 16 power generating plants located along the Chattahoochee River. And agriculture. There are around 573 agricultural water withdrawal permits that have been issued in the river basin. In my town, the closest industrial impact is the Smurfit West Rock Paper Mill, located in Cottonton, Alabama, on the western side of the river, just north of my collection site. This site produces southern yellow pine lumber and paper packaging like cardboard. Regarding water quality, the Environmental Protection Agency, EPA, has recently published a water report in 2024 that found E. coli in this section of the Chattahoochee River. PCBs were also tested for, but were not found in fish tissue. This is important to talk about because of the fact that if you look in the top, you can kind of see that is the one of the retention ponds for the plant. And these factors all contribute to pollution, and that can cause issues within my sample. Weather conditions of sample site. Another important factor to discuss is the weather conditions. So the date of my collection was on 4/10/2025. The time of day was 4:45 p.m. The weather was sunny. Temperature was 77. The daily low was 46. And the humidity was 33%. The barometric pressure was 30.08 inches of mercury. And wind was west-northwest 9.3 miles per hour. The notable weather. So thunderstorms and rain on 4/7/2025. Thunderstorms started on April 6th at 3:27 p.m. and the rain stopped after 10:51 p.m. on April 7th. The sample was collected within 72 hours after a rain event. This is notable because rain event sampling can increase the concentrations of pollutants in bodies of water compared to routine sampling. It is recommended to avoid contact with water sources within 72 hours of a rain event to prevent contact with poor water quality. The rain event could have significantly impacted water quality including increased pollution and higher turbidity. This is important to note when comparing the two jar samples. Methods and materials, site location. So if you look to your right, you can kind of see at the top picture, this is just an overview of the sample site on the Chattahoochee River. This is actually an interesting place. So this used to be where the log trucks used to be loaded on barges and taken to the paper mill before they built the bridge that now is used. But if you look at the bottom right picture, you can see where I sourced my plants. Right there in the bottom right in the red, that is where I got all of my alligator weed, all of my water, all of my duckweed, and then of course the algae was just present in the water. And in the yellow circle, that is where I got all of my soil. So the water condition had high turbidity. Like I said, plants sourced in red were alligator weed, duckweed, and algae, and the soil was sourced in yellow was mostly sandy clay and leaf litter. So some important things, the autotrophs that I collected, alligator weed, Alternanthera phylloxeroides. It is an invasive weed native to South America, according to the U.S. Fish and Wildlife Services. It is a fast-growing perennial amphibious herb that roots in soil near the edge of the water and extends hollow stems with roots over the water surface. It produces vegetatively through fragmentation of roots and stems. The reason I selected this species in particular was because of the rooting zone. I wanted to ensure that I had plenty of chances to catch as many microorganisms as possible. Now, common duckweed was my other one, Lemna minor. They are a small plant that has a single flat oval leaf that floats on the surface water. Fertig, no date. They primarily produce asexually by forming new stems from vegetative buds. This species can produce quickly, and it is an important food source for aquatic species. I selected those because I figured they would multiply pretty quickly. Algae, these are photosynthetic organisms found in many habitats. These are important primary producers of organic matter and oxygen for aquatic life. I'm unsure of the species of algae in my microcosm, but their presence is noticeable and it's naturally occurring. Methods and materials. So these are the materials. I used a half-cup measuring cup to collect soil. The kimchi jar was 4 by 4 by 7 inches with a volume of 87.96 squared inches. You can see that on the left in the picture. And the pickle jar, which was my second microcosm, was 5.5 by 5 by 6 inches with a volume of 117.81 squared inches. I used a disposable cup to collect my autotrophs. These were primarily duckweed and alligator weed, and I included a lot of root materials from the alligator weed to again increase the likelihood of capturing heterotrophs. A digital humidity and temperature gauge was used to monitor environmental conditions. The temperature stayed between 60 to 80 degrees Fahrenheit as it was indoors. For natural sunlight, this was provided by a north-facing windowsill to decrease the likelihood of increased water temperature by limiting exposure to direct sunlight. Observations and data, sunlight conditions. So this chart is called Optimal Sunlight Conditions based on cloud coverage. It was made in Excel from a compilation of weather data that I sourced from the website Day and Time. This graph shows the optimal sunlight conditions for each day with 12 total hours of daylight per day. Hours that contain rain, overcast, or mostly cloudy conditions were estimated to be less than 50% of optimal conditions, and hours with partial cloudy conditions or passing clouds were estimated to be about 50% of optimal conditions. Lastly, hours without any clouds would be estimated to be 100% optimal conditions, full sunlight, so 100% optimal conditions. So looking at this chart and data is important because there were more days that had less than optimal sunlight conditions. And less than optimal conditions would reduce the productivity levels of the autotrophs within my microcosm. So you'll see this chart repeated in the next few slides. It's a good reference to kind of get an idea of what's going on. Just keep in mind to look at this as well as the pictures. Observations and data, the microcosm on 12 April. So using the graph from the previous slide, let's take a look at our two microcosms and compare them to optimal sunlight conditions. So 12 April was the first day that my microcosm was actually observable. Before that, it was way too cloudy. Because remember, the rain event made it very, very cloudy, high turbidity. So sunlight was optimal on this particular day, on April 12th. The turbidity was high, making it difficult to observe any kind of macroinvertebrates. Algae was also increased, or at least appears to be, unless the greenish yellow water is just sediment that has not settled. I couldn't really distinguish between algae and sediment. And then, like I said, a few macroinvertebrates were observed, like mosquito larvae. Observations and data, microcosm on 19 April. So sunlight conditions were optimal. Turbidity was still slightly high. Algae was noticeable, mostly in the pickle jar, and more macroinvertebrates were observable. If you look at the picture, you can see this is definitely way more clear than the previous one. So at this point, I was starting to see a lot more macroinvertebrates in the kimchi jar. Observations and data, microcosm on 25 April. So here, again, sunlight was suboptimal based on our graph. So turbidity was decreased as the sediments have started to settle. Algal growth was pretty low, and the pickle jar seemed to have lower turbidity than the kimchi jar at this point, which is kind of interesting. So if you look at the picture, the kimchi jar looks a little bit darker than the pickle jar, but just keep that in mind. Like I said, always compare the two. Observations and data, microcosm on May 4th. So sunlight was increased on this date following suboptimal conditions before declining by May 5th. So this may have affected plant growth and algal growth. Turbidity was low in the kimchi jar, but high in the pickle jar. The algae was lower in the kimchi jar compared to the pickle jar, and there were less macroinvertebrates that were found in the pickle jar, while the kimchi jar had a lot of macroinvertebrates. And you can see the clear difference. The kimchi jar is a lot more clear than the pickle jar. So observations and data, the microcosm on May 17th. Here you can see extreme differences between the two microcosms. Sunlight on this day was fairly optimal with more hours at 100% compared to the overall trend of less than optimal conditions. That's probably what fueled the algal bloom on this particular day. So the turbidity in the kimchi jar is low, and you can actually see through the plant material. But however, in the pickle jar, it is extremely high turbidity. And the algae in the kimchi jar has increased, but it's still low compared to the pickle jar. You can kind of see the algal growth at the top of the pickle jar and kind of around the root zone. This is following, kind of decay, and I'll talk about this a little bit later. The pickle jar has a huge amount of algal growth, again, contributing to lower turbidity. Less macroinvertebrates were found in the kimchi jar, but they were still present, and the pickle jar had no macroinvertebrates. I could actually see dead worms in their tunnels in the soil. They weren't moving, so definitely they died. So observations and data, macroinvertebrates. So many different macroinvertebrates were observed. These populations represent the heterotrophs. So the main ones were amphipods in the form of scuds, aquatic worms, a diving beetle, a diving spider, a left-hand snail, mayfly larvae, and even mosquito larvae. Major findings in the kimchi jar. So in the kimchi jar, for the alligator weed, oh yeah, sorry. This photo was taken on 25 April to use for reference. But the alligator weed, the roots, the stems, and the leaves of it, they look pretty healthy and full. And the duckweed, the roots and leaves look pretty healthy again. There's not really a lot of algae, not a whole lot of growth of algae. Most of it is actually kind of concentrated on submerged plant stems, leaves, and roots. And as far as macroinvertebrates, the population is extremely high. There's plenty of individuals with many different species that were observed, but overall plant health looks really good, um, and the water is extremely clear. So again, major findings in the kimchi jar. This is to compare the two. So this was, the plant decline. The day this photo was taken is on 17 May. And if you kind of look at it, you can see clear difference. The plants are definitely declining, starting to break down. So the alligator weed, the submerged plant parts have clearly declined. They're starting to decay. Nearly all the duckweed has pretty much died off, and algae has begun to bloom towards the top of the microcosm. So this here, in this case, the algae is becoming the main autotrophs since the duckweed is missing. As far as macroinvertebrates, the population has definitely declined with less individuals. Some of the larger individual species have survived. I noticed a large snail that was observed consuming submerged roots and stems. There are also larger predatory macroinvertebrates that have been observed that were hunting within the root zone of the alligator weed, and those include a diving spider that you could actually see it had created a network of webs in the upper zone of the microcosm and above and below the water surface. So there was actually spider webbing in the water and above the water, and he kind of hung out at the top of the jar, but he would go down into the root zone to hunt. Major findings, macroinvertebrates of the kimchi jar. So there was a major relationship between the populations of autotrophs and heterotrophs. So this left graphic is basically just showing that relationship, how they all affect each other. They're interconnected. And then the right side kind of explains what I observed in order. So initially, macroinvertebrate populations of heterotrophs were very large in the kimchi jar, and then they consumed the plants and the algae, which were the autotrophs, at an extremely fast rate. The population of macroinvertebrates was very diverse and even included prey and predator species. So this is a good example of tertiary levels where the algal and plant populations represent the first trophic layer. Their primary consumers are the macroinvertebrates of the second trophic level. And then finally, the predators, like the spiders, they're the third trophic level. So initial observations showed an increase in heterotrophs as the turbidity decreased. So they were probably present, but I couldn't see them. So as that sediment was settling, I could really see them. These autotrophs consumed the plants and algae, decreasing those populations. The third trophic level decreased the populations of the second trophic level organisms by hunting. That was indicative by the spider web. This was most noticeable by the loss of mosquito larvae. So in the very beginning, I had a lot of mosquito larvae, and then they were just completely gone. So clearly, they were victims of predation. As a population of the second trophic level macroinvertebrates decreased, the algal levels began to increase towards the end of the experiment. So you can kind of see in that graphic on the right, as heterotrophs increased, plant growth decreased and algal growth decreased. But as the heterotrophs decreased, plant growth continued to decrease while algal growth increased. So this one here is major findings of the pickle jar. So like I said, for my experiment, I included a second microcosm, which was a pickle jar. I did this initially as a backup microcosm, and it ended up being pretty interesting. So I set the kimchi jar up with the first segment of alligator weed. So that one had a lot more root growth, and the pickle jar was the lower segment of alligator weed, and that had less root growth. There were very little macro invertebrates found in the pickle jar, with the exception of a few aquatic worms. And the absence of heterotrophs in the system led to the uncontrolled algal growth and duckweed growth. So the excess nutrients from the previous storm could have been a contributing factor for the algal bloom. So these plants probably were able to consume lots of nutrients from all that storm conditions, plus, I had stated some of the water quality issues. There'd be extra nutrients from all the water activity, urban activity, the E. coli present, different things. The pickle jar ended up being a good control to compare to the kimchi jar, where the kimchi jar had more of an abundance of heterotrophs. This jar represents the extreme imbalance of an ecosystem, where the first trophic level of autotrophs are left unchecked, and a crash in the system by causing a decline of insufficient populations of secondary and third trophic level organisms. So basically what I'm saying here is the algal bloom caused decreased dissolved oxygen, because as that algal bloom died off, it basically used up all the oxygen in the system, and that killed pretty much all of my heterotrophs and decomposers like the worms. So other than plant life, there was no life. So major findings, water quality. This is related to one of my hypotheses. So in this one, biotic sampling of waterways is a good way to test water quality using macroinvertebrates, because macroinvertebrates are good indicators of water quality since they have different tolerance levels of chemical, physical, and biological water conditions, such as water temperature, turbidity, pollution levels, and levels of dissolved oxygen. So macroinvertebrates are also lower organisms in the food chain that larger species depend on, and usually healthy water systems should be abundant in these organisms. So each group of macroinvertebrates can be represented by tolerance levels, with some being less tolerant to pollution. And that's shown, if you look at the right side, that's shown by this South Carolina adopt-a-stream image. You can see that there's definitely the top species at the very top, they are less tolerant of pollutants. So one of those includes the mayfly larvae. They are one of the macroinvertebrates that are intolerant of pollution. That's important to note. If you look at the top right graphic, that right there is most likely a mayfly larva. So that's important because in the kimchi jar, there was presence of mayfly larvae at the end of the experiment, which might indicate a healthy ecosystem, despite the initial assumption of poor water quality that was associated with water sampling following a rain event and initially low turbidity. So despite all that factors that were working against water quality, the rain, and all the pollution, we were still able to find a sensitive organism in this particular jar. So that's kind of a good indication that water quality has been improved in this jar. So for the pickle jar, the complete lack of macroinvertebrates in the pickle jar show low water quality. This can be attributed to either the presence of pollution or low levels of dissolved oxygen following the decomposition of plants and algae. And you know, the alligator weed was actually nearly completely decomposed. So as that decayed, that probably contributed to the low levels of dissolved oxygen. But considering that jar has mostly just algae and alligator weed, we can assume the water condition is not suitable for aquatic life. Well, aquatic life in the form of heterotrophs. All right, major findings, floating roots. I found that these are a primary habitat, and I'm going to play this video so you can kind of see, if you pay attention, you can kind of see some biological activity, including right in the middle. If you look closely, you'll be able to see there is a scud. So we're going to play this. And if you look at that kind of blackish stick in the middle, he's kind of like right on that. So the primary habitat within my system was the root zone of the alligator weed. The majority of my macroinvertebrates were found within this habitat. This habitat contained the most observable activity, including active feeding of root material and other organic material in this zone by different heterotrophs, like the snails and the scuds. There were also indications of predation by observing the diving beetle and the diving spider. They were extremely active in this zone. Additionally, aquatic worms, which are decomposers, were observed using this root network to increase their mobility, possibly to access nutrients within this zone. These were primarily found in my second most productive zone, though, which was the soil, where they created noticeable tunnels. And I'll show that in the next slide. But although alligator weed is an invasive species, this observation highlights the importance of floating aquatic plants and their roots as a primary habitat for aquatic species. It would be preferable to have native species like American water lily to fulfill this function, but I didn't sample a water lily. What I had access to was, unfortunately, an invasive species. So major findings, the soil zone. This was another primary habitat. So the soil in my microcosm, again, was the second primary habitat in my system, and it was a major storage for organic matter. This area was actively used by my most noticeable decomposers, which were aquatic worms. If you look in this picture, you can kind of see them hanging out in the bottom right corner. In this zone, again, you can see their extensive network of tunnels. You could see the buildup of sediments and other decomposed organic material, this kind of yellowish, powdery stuff on that leaf at the top, in the top middle area. It was definitely more noticeable in the last few weeks as plant materials decreased and organic matter increased on the surface of the soil and within the tunnels of aquatic worms. So major findings. Did my observations support my hypotheses with water quality and plant growth? So for water quality, my hypothesis was water quality and clarity will improve in 8 weeks as sediments settle and plants and animals sequester nutrients in the water. Water quality did improve in the kimchi jar, where there was an abundance of macroinvertebrates to provide the different vital niches in an ecosystem. In the kimchi jar, it contained autotrophs, heterotrophs, and decomposers. However, in the pickle jar, water quality initially improved, but decreased as algal growth was left unchecked. This kind of highlights the importance of different species to fill fulfill different biotic niches. As far as plant growth goes, my hypothesis was plant growth will increase with access to sunlight and an abundance of nutrients present in the water. This was definitely supported by the plant growth that increased in the pickle jar. The abundance of nutrients following a rain event was beneficial to plant growth in addition to access to sunlight. However, in the kimchi jar, plant growth only increased above water. Basically, the leaves of the alligator weed were the only ones that were not victim to the snail. The snail ate most of the other parts. The abundance of heterotrophs might have been too high for the system to support long term. This was evident by the overconsumption of plant materials. Plant growth could not keep up with the consumption by heterotrophs. Mostly the snail. He ate pretty much all of my plants. So in conclusion, there were three concepts that were clearly evident in this project. Concept 1, population growth. Concept 2, population dynamics. And concept 3, ecosystem, ecology, and biogeochemical cycles. So the concept of population growth was clear in both of my microcosms. In the kimchi jar, the population of heterotrophs declined as predation increased, and the population of autotrophs decreased. In the pickle jar, access to sunlight and nutrients allowed the population of plants and algae to increase, but the lack of heterotrophs caused the autotrophs to be overproductive. That was indicated by the algal bloom. The concept of population dynamics was also evident in the relationships between autotrophs and heterotrophs in the kimchi jar, where plants absorb nutrients and sunlight, making them available to heterotrophs that consume the plant materials. These heterotrophs pass those nutrients onto the predators that consume them, and then the decomposers, which were aquatic worms, broke down nutrients and waste from both plants and heterotrophs and stored them as organic matter in the soil. Ecosystem, ecology, and biogeochemical cycles were also represented in my microcosm. As I stated earlier, the autotrophs captured energy from the sun and passed that energy to heterotrophs and decomposers. With more time, the energy that was stored in the soil by the decomposers would be taken up by the plants and repeat the cycle. So why are microcosm studies relevant? Well, they're relevant because they allow observations of aquatic ecosystems. Many of the organisms in an aquatic ecosystem are just too small to regularly observe in their natural habitats. Plus, there's the nature of water, which is dynamic. Water is constantly flowing with new biotic and abiotic factors constantly changing or being introduced into the system. This makes it difficult to observe aquatic ecosystems. A microcosm is a much smaller environment that is much easier to control. You can limit the biotic and abiotic factors by controlling sunlight, temperature, and by containing the system. This type of study facilitates cause and effect analyses. You can easily manipulate the microcosm by creating near perfect conditions and then introducing biotic or abiotic factors in a controlled setting. For example, if you wanted to study the effects of fertilizer, you could set up multiple microcosms with nearly perfectly identical settings, like the same water, same organisms, same temperature, same light conditions, etc. You would basically inoculate a few of them with fertilizer and keep a few as controls by not introducing fertilizer. This would basically allow you to study how fertilizer interacts with different species of the microcosm. Basically, you'd be able to observe them by comparing the controls and the inoculated microcosms. Further research. So what further hypotheses would I like to test? I would like to test the importance of macroinvertebrates in an ecosystem. My hypothesis would be something like a balance of macroinvertebrates is necessary for a proper ecosystem to function. And to do this, I would have to try to capture many different macroinvertebrates to fulfill all the different niches. and I would probably need to do this with multiple microcosms for testing. And as far as what would I do next time to improve my project, to improve the project, I would include more microcosms, and the end results between my two samples were sourced from the same site. They were very different from one another. That basically shows why it's important to have more sampling data for experiments. So more microcosms, more data, a lot more comprehensive to improve my findings. And these are my resources, and they continue on to the next slide.